This is chapter 26. We're going to go over fluid, electrolyte, and acid-base homeostasis. Okay, so in adults, body fluids make up between 55% and 60% of the total body mass in females and males respectively. Now, body fluids are present in main compartments. So these main compartments are going to be inside of cells and outside of cells. So about 2/3 of body fluid is inside of cells. So this is going to be intracellular fluid. Intracellular meaning inside of cells. And then the other 1/3 is called extracellular fluid. And this is going to be outside of cells. And this includes pretty much all other body fluids that isn't cytosol inside the cells. Of the extracellular fluid, about 80% is interstitial fluid. So this is going to occupy the microscopic space between tissue cells. And then the other 20% of extracellular fluid is plasma, which is the liquid portion of blood. So the body is in fluid balance. So this is when the required amounts of water and solutes are present, and they're going to be correctly proportioned among the various compartments. Now, water is by far the largest single component of the body. It makes up between 45 and 75% of total body mass, depending on age and gender. So here in this diagram, we can see the body fluid compartments of a female on the left and a male on the right. We can see how much makes up intracellular fluid and extracellular fluid. So here with the females, we have 55% of the body makeup is made up of fluids, whereas in males, about 60% is fluids. And of the total body fluid, we can see the 2/3 being that intracellular fluid. Remember that fluid inside the cells, so cytosol. And then 1/3 of the fluids being extracellular fluid. And then of that extracellular fluid, we have another diagram here being that 80% is interstitial fluid and 20% making up plasma in blood. Two general barriers separate intracellular fluid, interstitial fluid, and blood plasma. The first one, right here, we're going to label that one, is going to be the plasma membrane. That's our first barrier here. And so this is the plasma membrane of individual cells, and it separates intracellular fluid from the surrounding interstitial fluid. Now, we learned in chapter 3 that the plasma membrane is a selectively permeable barrier. So what that means, remember, is that it allows some substances to cross, but it blocks the movement of other substances. Now, in addition, active transport pumps will work continuously to maintain different concentrations of certain ions in the cytosol versus the interstitial fluid. The second barrier that we have right here are the blood vessel walls. So blood vessel walls will divide the interstitial fluid from the blood plasma. Only in the capillaries, which remember are the smallest blood vessels, this is where the walls are thin enough and leaky enough to permit the exchange of water and solutes between the blood plasma and the interstitial fluid. So the processes of filtration, reabsorption, diffusion, and osmosis all allow continual exchange of water and solutes among the body compartments, but the volume of fluid in each compartment really remains remarkably stable. Here we can see on this picture on the left, we can see those two barriers. So here inside the cells, we have this intracellular fluid. Just kind of label this green here. So this is going to be that intracellular fluid, that's cytosol, inside the cell. So here is our first barrier, the plasma membrane. So this is going to separate that intracellular fluid inside the cell from the interstitial fluid surrounding the cell and kind of going around all these other cells. So that's the first barrier there. And that interstitial fluid will come all the way up to here, because this is where we're going to see our second barrier, which I'll label this here in dark blue, which is the wall of the blood vessel. So it's going to be our second barrier, and it's going to separate that interstitial fluid in light blue there from the blood plasma, which I'll just label in here. So it's going to be the blood plasma inside the blood vessel there. So we have those barriers separating the fluids in different compartments. Now, because osmosis is going to be the primary means of water movement between intercellular fluid and interstitial fluid, the concentration of solutes in these fluids really determines the direction of water movement. Now, because solutes in body fluids are electrolytes, so what this means is that they're inorganic compounds that can dissociate into ions, fluid balance really is closely related to electrolyte balance. Now, because the intake of water and electrolytes, remember ions, rarely occurs in exactly the same proportions as their presence in the body fluids, the ability for the kidneys to excrete excess water by producing dilute urine, or to excrete excess electrolytes by producing concentrated urine, is really one of the most important things in the maintenance of fluid homeostasis. So the body can gain and lose water in a variety of different ways. Here on the left we have how we gain water throughout the day, and on the right is how we lose water. So the body can actually gain water by ingestion and by metabolic synthesis. Now, the main sources of body water are going to be ingested liquids, about 1,600 milliliters, and also moist foods. This is where water is going to be absorbed from the gastrointestinal tract. Now, these two ingested waters total about 2,300 mils per day. Now, these numbers you don't specifically have to remember, it's just for interest's sake. Now, the other source of water is going to be metabolic water. Now, this is water that's produced in the body mainly when electrons are accepted by oxygen during aerobic respiration. A smaller extent is going to be produced during dehydration synthesis reactions as well, but mainly it's going to be during that aerobic respiration. And that metabolic water accounts for only about 200 mils per day. So daily water gain from these two sources, from ingestion and metabolic synthesis, totals about 2,500 mils per day. Now, normally your body fluid volume remains pretty constant because water loss is going to equal water gain. Now, water loss can occur in 4 ways. Now, each day, the kidneys are going to excrete about 1,500 mils in urine. The skin is going to evaporate about 600 mils. The lungs exhale about 300 milliliters as water vapor. And then the gastrointestinal tract is going to eliminate about 100 mils in feces. The amount of water lost by a given route between these 4 routes is going to vary considerably over time, though. The volume of metabolic water formed in the body depends entirely on the level of aerobic respiration. And this is going to reflect the demand for ATP in body cells. So when more ATP is produced, subsequently more water is formed. Now, body water gain is regulated mainly by the volume of water intake or how much fluid you drink. Now, an area in the hypothalamus, we call it the thirst center. This is going to govern your urge to drink. When water loss is greater than water gain, dehydration is going to stimulate thirst. So dehydration is going to be a decrease in volume and an increase in the osmolarity of body fluids. Okay, so let's talk about dehydration a little bit. So this diagram here shows us the pathway through which dehydration at the top is going to stimulate thirst near the bottom here. Now remember that dehydration occurs when water loss is greater than water gain. Okay, so first we're going to cover our blood volume route here. So when body mass decreases by 2% due to fluid loss, we're going to get mild dehydration occurring. So a decrease in blood volume is going to cause blood pressure to fall. Now this change stimulates the kidneys to release renin, which is going to promote the formation of angiotensin II. So this is going to stimulate the thirst center in the hypothalamus. In addition to increased nerve impulses from osmoreceptors in the hypothalamus. So this is going to stimulate those osmoreceptors. Again, these increased nerve impulses from the osmoreceptors, as well as the increase in angiotensin II in the blood, are both going to stimulate that thirst center in the hypothalamus. Another signal that can stimulate thirst is going to be the neurons in the mouth detecting dryness due to a decreased flow of saliva. So we get that dry mouth and pharynx, which is also going to stimulate the thirst center. Now, as a result, the sensation of thirst increases, which usually increases water intake, bringing us back to homeostasis. Even though the loss of water and solutes through sweating and exhalation increases during exercise, the elimination of excess body water or solutes really occurs by controlling their loss in the urine. The extent of urinary salt loss is the main factor that determines our body fluid volume. So the main reason for that is that water follows solutes in osmosis. The main solutes in extracellular fluid and in urine are going to be sodium ions and chloride ions. Now, in a similar way, the main factor that determines body fluid osmolarity is the extent of urinary water loss. So this is going to be a really important thing to remember, that wherever solutes go, water follows. So this is important. Because our diet contains a highly variable amount of sodium chloride, urinary excretion of sodium and chloride must also vary in order to maintain homeostasis. Hormonal changes regulate the urinary loss of these ions, which in turn affects blood volume. Now here we have a diagram that shows the hormonal regulation of renal sodium and chloride reabsorption. So these would be the changes that would occur right after a salty meal, for example. So we have an increased intake of sodium chloride at the top here. Now this produces an increase in plasma levels of sodium and chloride. This is in the blood. And remember that sodium and chloride are the major contributors to osmolarity of extracellular fluid. So as a result, the osmolarity of the interstitial fluid is going to increase, and this causes a movement of water from the intracellular fluid to the interstitial fluid and then to the plasma. So remember, water follows solutes. So we have this increase in sodium and chloride in the plasma. So then we're going to have this increase of osmosis from the intracellular fluid and then to the interstitial fluid and then to the blood plasma where those excess ions are. So this water movement is going to increase blood volume overall. Now, recall that the three main hormones that regulate sodium and chloride reabsorption and therefore the amount lost in urine are angiotensin II, aldosterone, and the atrial natriuretic peptide, or ANP. So these are going to be the three main hormones that regulate renal sodium and chloride reabsorption. So when we go back to the diagram, there's a couple things that happen when blood volume increases. So first off, we have this increased stretching in the atria of the heart, and this causes increased release of ANP. So we'll go, well, I guess here this causes this reduced reabsorption by sodium chloride in the kidneys. Okay, let's go back up here. This increased blood volume will also cause this decreased release of renin by those juxtaglomerular cells, resulting in a decreased formation of angiotensin II and a decreased release of aldosterone, as well as an increased glomerular filtration rate, both of which are going to contribute to that reduced reabsorption of sodium chloride in the kidneys. So if we're not going to be reabsorbing as much sodium chloride in the kidneys, this is going to result in an increased loss of sodium and chloride in the urine. So if we're going to lose more of that sodium and chloride in the urine, or more sodium and chloride is going to stay within the urine, water is going to follow, remember. So we're going to get an increased loss of water in urine by osmosis, and as a result, this is going to decrease blood volume and get us back to homeostasis. So like I just went over in the last slide, the three most important hormones that regulate the extent of renal sodium and chloride reabsorption are going to be angiotensin II, aldosterone, and atrial natriuretic peptide, or ANP. So when your body is dehydrated, so say we have like low water in our body, angiotensin II and aldosterone will promote the urinary reabsorption of sodium and chloride. So we're going to get an increase in sodium and chloride for both of these. So this is reabsorption, so where you're holding on to these ions instead of releasing them into the urine. And as a result, we're also going to be reabsorbing water by osmosis, because remember, water is going to follow these ions. Now, on the other hand, if we have like excess water, so this excess water is going to cause an increase in blood volume. So we might see this, or this might occur after you have like a supersized drink or something like that. We have an increase in blood volume, and this is going to cause the atria in your heart to stretch, and this is going to promote the release of ANP. Now, ANP promotes natriuresis. So this is natriuresis is elevated urinary excretion of sodium and chloride, which is followed by water excretion. So this is going to decrease blood volume. So this is going to be decrease in sodium, chloride, and water. Now when I say decrease, I'm just saying your body decreases in these ions because we're going to be excreting it in the urine. So when we decrease in water, this also decreases blood volume. Now, the major hormone that regulates water loss is the antidiuretic hormone, or ADH. Now, this hormone is produced by the neurosecretory cells that extend from the hypothalamus to the posterior pituitary. Now, in addition to stimulating the thirst mechanism, an increase in the osmolarity of blood fluids stimulates the release of ADH. Now, this increase in ADH promotes the insertion of these water channel proteins. So remember, these are aquaporins. So higher ADH, so if we increase ADH, this is also going to increase the insertion of aquaporins. So aquaporin 2. And this is going to increase those insertions in the apical membranes of the principal cells in the collecting ducts of the kidneys. So we're going to have a lot more of these aquaporin proteins in the collecting ducts in the kidneys, and as a result, the permeability of these principal cells to water is going to increase. So this is going to increase water permeability. So this increase in water permeability is going to increase water reabsorption. So we're going to be holding on to more water and not getting rid of as much water in our urine. So instead of a very dilute urine, when we hold on to a lot of water, the urine is going to be more concentrated because the urine contains less water. Here's a really nice table that summarizes the factors that maintain our body water balance. So this is a really good one to look over. So we have things like the angiotensin II, aldosterone, ANP, and ADH. We're all here, and it shows the mechanism and also the effect that these hormones have. Again, these are all things that we've talked about already, but just having a nice table to kind of summarize all of that here. So definitely take a chance to look this over, and you can use this in your studying. When a person steadily consumes water faster than the kidneys can excrete it, or when renal function is really poor, the result may be water intoxication. So this is a state in which excessive body water causes the cells to swell dangerously, and sometimes these cells don't survive. So here we have kind of a figure that shows the series of events that would occur in water intoxication. So we have water intoxication here at the bottom, so we're kind of going to go over the steps that would lead to that. So at the top here, if there's excessive blood loss, excessive sweating or vomiting or diarrhea, and this fluid loss is replaced by drinking plain water, so this is going to be water without ions, water with no electrolytes or anything, then the body fluid becomes more dilute. So essentially you're losing a lot of fluid that has ions and electrolytes in it by sweating or vomiting or diarrhea or blood loss, and you're replacing it with very dilute water that doesn't have any ions in it. And so your body is going to try to compensate for that. So this is going to result in that decreased sodium concentration in the interstitial fluid and the plasma. It's going to fall below normal range. And when the sodium concentration of interstitial fluid decreases, its osmolarity also falls. So we have that decreased osmolarity. Now, because your body's going to try to maintain homeostasis across all the compartments or all the fluid compartments in your body, the net result is going to be osmosis of water from the interstitial fluid into the intracellular fluid, so into the cytosol of the cells. So we're trying to balance out the osmolarity between all the fluid compartments in the body. So we're having an excess amount of water going into our cells. So water entering the cells is going to cause them to swell. And this is an unnatural thing. And this is where we have that water intoxication happening. Now, this can produce convulsions, coma, and even possibly death. So drinking plain water without any ions in it after you've lost a lot of fluid by even just sweating and exercising a lot it can be quite dangerous. So we're going to look a little bit more into electrolytes in the body. So ions that are formed when electrolytes dissolve and dissociate have 4 general functions in the body. So the first one is that because these ions are largely confined to particular fluid compartments and they're a lot more numerous than non-electrolytes, certain ions can actually control the osmosis of water between fluid compartments. So they're actually controlling the movement of water between different compartments. Ions also help to maintain the acid-base balance that's required for normal cellular activities. Ions carry electrical current as well, so this is really, really important. This allows for the production of action potentials and graded potentials. So these ions that are produced when electrolytes dissociate are very important for our everyday lives. Several ions also serve as cofactors. So these are ions that are needed for optimal activities of enzymes. Again, a very important role. To compare the charge carried by ions in different solutions, the concentration of ions is typically expressed in units of milliequivalents per liter. Now these units give the concentration of cations or anions in a given volume of solution. Blood plasma, interstitial fluid, and intracellular fluid all have different concentrations of electrolytes and protein ions. Now blood plasma contains many protein ions and interstitial fluid contains only a few, but we do have a diagram we're going to look at these differences between the different fluid compartments. So this graph here compares the concentration of the main electrolytes in protein and ions in blood plasma in red, interstitial fluid is in blue, and intracellular fluid in yellow. Now, one main difference that we see between the two extracellular fluids, remember this is going to be the blood plasma and interstitial fluid, is that blood plasma contains more protein anions in contrast to the interstitial fluid, which has very few. So we can have 20 milliequivalents per liter in that blood plasma versus 2 milliequivalents per liter of protein anions in the interstitial fluid. The electrolytic content of intracellular fluid differs quite a bit from that of the extracellular fluids as well. In extracellular fluid, the most abundant cation is sodium, and the most abundant anion is chloride. So we can see the red and blue for those two extracellular fluids are highest in the sodium and chloride compared to the intracellular fluid levels here. Now in intracellular fluid, the most abundant cation is potassium. Let's see, much, much higher here than in the extracellular fluids. And the most abundant anions are going to be proteins and phosphates. So in the next couple slides, we're just going to be going over these ions. First off, we have sodium. This is the most abundant ion in extracellular fluid, and it is used for a lot of things, as we've kind of talked about all year, including impulse transmission. Think about action potentials, also in muscle contraction, and fluid and electrolyte balance. Now remember that sodium levels are controlled by aldosterone, antidiuretic hormone, and atrial natriuretic peptide. Next we have chloride. this is the major extracellular ion, really helps to regulate osmotic pressure between compartments. It does form hydrochloric acid in the stomach, and it's mainly controlled by aldosterone. Next, we have potassium. This is going to be the most abundant cation in extracellular fluid. It's also involved in action potentials, in fluid volume regulation, also involved in muscle contractions, and also in regulating pH. Mineralocorticoids regulate plasma levels of potassium, so this is going to be mainly aldosterone. Then we have bicarbonate. This is also a very important plasma ion, and it's a major member of the acid-base buffer system. The kidneys will reabsorb or secrete bicarbonate for final acid-base balance. Now, because such a large amount of calcium is stored in bone, it's really the most abundant mineral in the body. About 98% of the calcium in adults is located in the skeleton and in the teeth. It's used for blood coagulation, for neurotransmitter release, even things like muscle tone and nerve excitability and muscle excitability. The most important regulator of calcium concentration in blood plasma is the parathyroid hormone, or PTH. About 85% of the phosphate in adults is present in calcium phosphate salts. Now, these are going to be the structural components of bone and teeth. Phosphate is also used as a buffer system, and it's also regulated by the parathyroid hormone and also calcitriol. In adults, about 54% of total body magnesium is part of bone matrix as magnesium salts, and the remaining about 46% occurs as magnesium ions in intracellular and extracellular fluid. So magnesium is going to activate enzymes. It's involved in carbohydrate and protein metabolism. It's also used for myocardial function, in the transmission in the central nervous system, so these action potentials, and also in operation of the sodium pump. Now on this slide and on the next one, we have a really nice table summary of the blood electrolyte imbalances. Now you won't have to memorize the effects of the deficiency or excess of these electrolytes for the exam or anything. This is more just for your own interest's sake. You can just kind of read it over if you want. On this slide, we have the electrolytes on the left. We have sodium, chloride, and potassium. Here we have their deficiencies and the excess of those electrolytes. And then on this slide, we have calcium, phosphate, and magnesium. Again, you don't have to memorize this for the exam, but just for your interest's sake. Okay, we're going to go over acid-base balance a little bit and how your body kind of maintains the acid-base balance of your fluids. So in general, the pH of your arterial blood has quite a small range. So we're really going to range from 7.35 to 7.45. And really your body tries to maintain the pH level within that range. It's very, very particular. Now, there are a lot of mechanisms that really help to maintain this pH. We will discuss each of these. So we're first going to go over buffer systems. Exhalation of carbon dioxide is another big one that helps to regulate the blood pH. And then also the kidney's excretion of hydrogen ions. So we're going to start out by talking about buffer systems. Now, in general, buffer systems really help to maintain pH levels so that when there's a buffer system in a solution, the pH will change very, very little when a small amount of strong acid or strong base is added to that solution. So in other words, when we're talking about buffer systems within our blood, we're talking about these compounds that will help to maintain the pH at a relatively very steady state, kind of regardless of whether there's acids or bases added to our blood. Now we're going to talk about three main buffer systems that our body uses in the blood. So the first one here is going to be the protein buffer system. Then the next one is carbonic acid bicarbonate buffer system. So carbonic acid, you can do a dash bicarbonate. And then the third one is called the phosphate buffer system. So these are going to be the three buffer systems that we will discuss in the next couple slides. So the first one we have here, which is that protein buffer system. So we have an end of a protein here. So really the main parts for this particular section that we need to talk about is we have an amine group. We've kind of talked about this a little bit when we looked over proteins in general, but here we'll just call this an amino group. So this is the amino group, and then this is the carboxyl group. Okay, so these are going to be mainly the one that we're going to look at here is going to be their carboxyl group, at least for this slide. So buffer systems, like I said, help to maintain a constant pH regardless of whether there's acids or bases added to a solution. So here with our protein buffer system, we kind of have our two main groups that are going to act differently dependent on whether there's acids or bases added. So in this case, we have an increase in pH. Now an increase in pH means that it gets more basic. And if a solution is more basic, that means there are fewer hydrogen ions in solution. So in order to bring us back to homeostasis, we need to essentially add hydrogen ions. And this is what this protein buffer system is going to do. So when we kind of get a shift in pH, even a little bit, buffer systems work to either add or remove hydrogen ions in order to kind of shift the pH back to normal. So here, this is where we have an increase in pH. This is where this carboxyl group here is going to dissociate. So that means right here, we're going to break this bond between the oxygen and the hydrogen, releasing a hydrogen ion into solution, and therefore we're going to decrease the pH because we're adding hydrogen ions, kind of returning us back to homeostasis that way. So this is one way that a buffer system works when there is a base added. So when the solution becomes more basic, a buffer will add hydrogen ions to solution in order to bring the pH back down and kind of counteract those effects of the base. So now we're going to see what happens when an acid is added to solution. So this is when kind of more hydrogen ions are added to your blood for whatever reason. So this is where we have acid added, and therefore it's going to be a drop in pH. So when there is a drop in pH, essentially we're just going to see more hydrogen ions in the solution. And in buffer systems, in particular with this protein buffer system, this is where we are going to see the amino group go to work. So we have these extra hydrogen ions, so we kind of need to mop them up. We need to take care of them, get those free-floating hydrogen ions out of the solution, and kind of essentially bond it to something so that they're not affecting the pH. So we have this amino group over here, and this is the side of the protein that's actually going to have the function when an acid is added to solution. So the nitrogen will have this extra lone pair of electrons, and sorry about that, this lone pair of electrons is going to bond to extra hydrogen ions in solution. So that amino group is essentially going to act like a base, and it's going to pick up a free-floating hydrogen ion. Now, each amino group on a protein is only going to pick up one hydrogen ion, so we're only going to see NH3s. We're not going to see anything like NH4 or NH5. It kind of breaks some chemistry rules. So we're only going to see NH3, and we're going to see that positive charge on the amino group. But what this does is that it kind of mops up those free-floating hydrogen ions, and as a result, the pH is going to increase because we're getting rid of those excess hydrogen ions in solution. We're bonding them to the amino group of the protein, and this helps to maintain a steady pH. So this is kind of how those proteins are going to act differently depending on whether there's an acid or a base added. So when there's an acid added, so this is just an acid added to solution, the protein buffer is going to have its amino group acting. And when we have a base added to the solution, to the blood, it's when the proteins will have its carboxyl group functioning. Okay. So here we kind of have an example of a real-life protein buffer system. So this is an actual system that we see in our blood. So kind of disregard these top two equations for a second. This is going to be the carbonic acid bicarbonate buffer system, which we actually will look at next. But right now what I want to draw your attention to here is just the bottom equation here. So we have an oxyhemoglobin. Actually, I'll just highlight these. So an oxyhemoglobin here, so this is just your hemoglobin, which is bonded to an oxygen molecule. So these are in the red blood cells, and at times we have just excess hydrogen ions that are floating around. In this case, they come from the previous equation up here from the carbonic acid. When they go through a reaction, then they release hydrogen ions. And so these hydrogen ions are from that reaction, but essentially right now they're just kind of free floating, bringing down the pH, making the blood more acidic. So your body doesn't really like that. We don't want to have acidic blood. So in this case, with a nice protein buffer as hemoglobin in the blood, that hemoglobin will dissociate from the oxygen molecule, and it will bond to the free floating hydrogen ions. So we are removing those hydrogen ions from the solution, bonding them to a protein, and kind of balancing out that pH. So when we have a free-floating hydrogen ion as in here, this is decreasing the pH level. And when we bond it to something else, like hemoglobin over here, we're removing that hydrogen ion from solution, increasing the overall pH level of the solution. So now we're going to move on to the second buffer system, just the carbonic acid bicarbonate buffer system. Now essentially all three of these buffer systems will kind of function the same way. When there's too many hydrogen ions or the solution's too acidic, these buffer systems will mop up the excess hydrogen ions. And when there's a base added or the pH of solution is too high, there's another part of that same buffer system is going to add hydrogen ions to solution. One thing I forgot to mention is that the buffer systems will always have a weak base that will kind of mop up excess hydrogen ions and a weak acid, which will release hydrogen ions into solution. So with our protein buffer system, we had our carboxyl group. This is just for the protein buffer system that we talked about before. The carboxyl group acts as a weak acid, and then our amino group is going to act as a weak base. So we can see those weak acids and weak bases as well in these carbonic acid bicarbonate buffer systems. So in this diagram, again, we're going to start out with if the solution is too acidic. So again, we have these excess hydrogen ions floating around in solution are causing the pH to fall in the solution. So in this case, we have those excess hydrogen ions. The bicarbonate ion, acting as a weak base, will bond to those excess hydrogen ions, or each bicarbonate ion will bond to one hydrogen ion, creating carbonic acid. So again, we're just going to be bonding to those free-floating hydrogen ions, removing them from the solution, and increasing the pH overall, just like we saw with the protein buffer system. Here on the other side, we have an increase in pH. So for example, a base was added. So this is when the carbonic acid can provide hydrogen ions to solution. So it will dissociate into a free-floating hydrogen ion and a bicarbonate ion, decreasing that pH, making the solution more acidic because we're just releasing those free-floating hydrogen ions back into solution. So again, like I said, these buffer systems are all essentially the same in the basics of it. If the pH rises, they're going to have a section that releases hydrogen ions into solution to increase the pH, bring us back to homeostasis. And on the other hand, if the pH falls and the solution becomes too acidic, then another part of the buffer system will mop up or bond to those excess hydrogen ions that are floating around in solution. And when those hydrogen ions become bonded to something, then it will increase the pH, making the solution less acidic. Now lastly, we have the phosphate buffer system. Now this one is slightly different, but again, at its base, it is the same acid-base homeostasis buffer system. So in these systems, we are kind of working between dihydrogen phosphate and monohydrogen phosphate. These are going to be our weak acids and weak bases. So here in this case, we have very few free-floating hydrogen ions in solution on the left of this equation. So this is going to be a very high pH. So we're looking at very basic solution. And so when the pH is pretty high, the dihydrogen phosphate will act as a weak acid, releasing one of its hydrogen ions here. And those hydrogen ions will often go towards the hydroxide ion here, bonding it and kind of making water. But with removing those hydroxide ions from solution, we are making the solution less basic as well. So even though we don't have exactly any more free-floating hydrogen ions on the right of this equation, by getting rid of those free-floating hydroxide ions, we are still decreasing the pH on the right of this equation here. So again, it's a little bit different, but we're still kind of had that basic understanding of releasing hydrogen ions when the pH is too high in order to drop the pH. And on the other hand, when the pH is too low, then we're going to bond to excess hydrogen ions that are free floating in order to mop them up and get them out of solution. And here we have kind of the other situation we have excess hydrogen ions, so we have a decrease in pH. And when we have a decrease in pH, this is when the monohydrogen phosphate, acting as a weak base, is going to come into play for this phosphate system, phosphate buffer system. And in this case, this is when the potassium here has those lone pairs, is going to bond to excess free-floating hydrogen ions, bond to them, remove them from solution, and turn into dihydrogen phosphate. Now, I guess you would have noticed that all of these buffer systems essentially are just reversible systems. So we can kind of draw a line this way or an arrow this way, depending on if the pH has dropped or the pH has increased. So buffer systems just are reversible depending on the pH of the solution. Now, another way that our body regulates the pH in our blood is by the exhalation of carbon dioxide. Now, this isn't a buffer system per se, but it does kind of act in a similar way. So when we have carbon dioxide in our blood from the metabolism of cells, when it mixes with water in the blood, it forms carbonic acid. Now, carbonic acid is going to dissociate into hydrogen ions and bicarbonate ions. Now, this kind of reversible reaction really kind of hinders on a very specific principle from chemistry. And this is Le Chatelier's principle. Sorry, Chatelier. So this is a principle that you may have learned in chemistry. Now, you won't have to know this principle for our class, but if you have taken chemistry, this may help you to understand kind of what's going on here. Otherwise, kind of disregard the name. It doesn't matter that much to us. But just if it may help some people in understanding these reversible reactions and kind of how this exhalation of carbon dioxide helps to regulate our blood pH. Now, Le Chatelier's principle essentially states that when one side of a reversible reaction increases, so say we have more CO2, it's going to shift it to the other side. So more CO2 is going to result in an increase in production of carbonic acid. And the same is true kind of going the other way, since it is a reversible reaction. The principle states that if we increase our carbonic acid, it's going to shift the reaction to the opposite side, increasing in CO2 and or water. So we're going to kind of use this principle to understand how carbon dioxide over here affects the concentration of hydrogen ions in the blood. Because at first glance, they're kind of unrelated, but I assure you that they are not. So we do produce a lot of carbon dioxide from metabolism, and so when we exhale CO2, we are decreasing the concentration of CO2 in our lungs and in our blood. So when we decrease that CO2 by exhaling, we are also shifting our reaction this way. So when we're shifting our reaction this way, we're having more carbonic acid that's being formed into carbon dioxide and water. So because more carbonic acid is being used to create carbon dioxide, we're decreasing the amount of carbonic acid because we're kind of using more. We can kind of think of it like a production line. For example, carbon dioxide over here are going to be tablets. Kind of the end of the production line where we have like an iPod, for example. Carbonic acid are going to be tablet screens. This is just a really simple example. It doesn't really fit for everything, but for my little example, it will. So when we sell sell more tablets over here, we're going to have fewer tablets in the store, and so we need more tablet screens in order to make tablets to replace the ones that we've sold. So for example, we had 100 tablets and we sold 50, so now we only have 50 tablets. And we had 100 tablet screens, but in order to replace the 50 tablets that we sold, we're going to use 50 tablet screens to produce more tablets and get us back up to 100. So we're decreasing the number of tablet screens because the number of tablets has gone down. Hopefully that kind of little example makes sense. So that's kind of what Le Chatelier's principle kind of looks at, just in very simple, brief terms. So back to our equation up here. When we breathe out more carbon dioxide, we're getting rid of that carbon dioxide, and so we got to use more carbonic acid to create more carbon dioxide. So that level of carbonic acid decreases because we are shifting our equation to the left here. Now, at the same time, because this is a longer equation, when we decrease our carbonic acid, we're also going to be decreasing stuff on the right side of this equation. Because decreasing this, we're kind of like creating a hole right here. So there's a hole here. So we're kind of got to fill that hole with stuff from the right side of the equation. So we're going to be using either more hydrogen ions or more bicarbonate ions in order to create more carbonic acid, because we're creating that hole of product, you could say, in the carbonic acid section. So when we decrease carbon dioxide, we'll cause a decrease in carbonic acid and subsequently a decrease in hydrogen ions. So this is kind of how those are related. So when we breathe out more carbon dioxide, the result is that we are also decreasing the amount of hydrogen ions in our blood because we're shifting these reactions to the left. So really the takeaway from this acid-base balance in carbon dioxide is that when we breathe out more CO2, or our exhalation of carbon dioxide is higher, this is going to result in a decrease of hydrogen ions, therefore an increase in pH, less acidic. Now, on the other hand, when we retain CO2, this is going to have an opposite effect, and we're going to go the other way. So then we're going to have an increase in CO2 is going to result in a shift of this equation to the right, resulting in an increase in carbonic acid. That's going to shift the equation to the right again and result in increase in hydrogen ions. So we'll just say keep in CO2. We don't breathe as much CO2 out. It's going to result in an increase in hydrogen ions and therefore a decrease in pH. So it's going to result in a more acidic blood. Now here we have a negative feedback diagram. It kind of shows the negative feedback mechanism for the regulation of acid-base balance when we exhale carbon dioxide. So at the top here, we have a decrease in blood pH. So this is an increase in hydrogen concentration. Again, that's a more acidic solution. And this is going to be detected by chemoreceptors, both in the medulla oblongata and also in the aortic and carotid bodies. Now these are going to send input or send nerve impulses to the dorsal respiratory group in the medulla oblongata, which is going to be the control center for this negative feedback loop. And dorsal respiratory group then is going to send impulses to the diaphragm, which is the effector in this group, which is going to contract more forcefully and frequently so that we have more carbon dioxide exhaled. So here we have this response. This is kind of just summarizes that equation that we saw on the last slide, that as less carbonic acid forms, we have fewer hydrogen ions present, and then the blood pH is going to increase, or hydrogen ion concentration decreases. It's going to become less acidic, and because of this, we're going to return to homeostasis. So that is kind of our real-world example of how forcefully exhaling more carbon dioxide is going to make our blood less acidic. Okay, now the last way that, or major way that our body is going to regulate acid-base balance in our blood is going to be the kidney's excretion of hydrogen ions. Now here we're going to come into another term, which is non-volatile acids. So a non-volatile acid is an acid produced in the body from sources other than carbon dioxide. So non-volatile acids cannot be excreted by the lungs by that carbon dioxide excretion mechanism that we just talked about. So this is where all those other acids are going to have to be excreted from the body because they cannot be breathed out through carbon dioxide, so the kidneys will have to get rid of them. So with this regulation mechanism, we're going to have cells in the proximal convoluted tubules and the collecting ducts, which will secrete hydrogen ions into the tubular fluid to excrete it in the urine. So here we can see a nice diagram of a magnified section of the collecting duct in the kidney. So we can see here these individual intercalated cells in the collecting duct. On the left is going to be the tubule lumen, and on the right we have the capillary. On the bottom right here we also have the key showing us what pictures the little proton pump is going to be representing, and also the bicarbonate ion chloride antiporter, and this is going to be in the basolateral membrane, and also this little dotted arrow representing diffusion. So we're going to start out here with carbon dioxide and water in the cell, just some of those products of metabolism. And we have carbonic anhydrase here. Remember that enzyme that's going to assist that rapid conversion of carbon dioxide and water into carbonic acid here. And that carbonic acid is going to dissociate into hydrogen ions and bicarbonate ions. Now with ATP, these proton pumps here in the apical membrane of the intercalated cells are going to pump those hydrogen ions out into the tubule lumen to be excreted in the urine. And then on the other side here in the basolateral membrane, I'll circle it actually, we have this bicarbonate ion chloride antiporter that's going to pump those bicarbonate ions back into the blood to be used as a buffer there. And then it's an antiporter, so it's just going to be pumping in one chloride for each bicarbonate ion to kind of maintain that balance, that electrochemical balance. So this is just how the kidney is going to get rid of excess hydrogen ions, because these are going to be from non-volatile acids. Now, because we don't want our urine to be extremely acidic also, that would cause problems in our bladder and our urethra, for example, the hydrogen ions that are pumped into the tubular fluid are also going to be buffered. So some of those hydrogen ions that are pumped out here, as we just went over in that last diagram, are going to be buffered by a couple of different ions here. Now first at the top here we have ammonia, and ammonia is also going to be able to bind to those excess free-floating hydrogen ions to produce ammonium. So ammonia is NH3, ammonium, NH4. Again, you don't have to know this specifically, but yeah, an ammonia buffer here. And then we also have a really nice phosphate buffer that will also bind to those hydrogen ions, or the monohydrogen phosphate to bind to the excess hydrogen ions to produce the dihydrogen phosphate. So this is essentially like the phosphate buffer system that we saw before in the blood. It is also a little bit going to be in the tubular fluid in the kidneys. So just going to be buffering those excess hydrogen ions that the intercalated cells pump into the tubular fluid as well. Again, so here is just a really nice table just summarizing everything we've covered so far in the past couple slides. We have the mechanisms that are going to be maintaining the pH in body fluids. Remember, that includes the buffer systems, the exhalation of carbon dioxide, and then the kidney excretion. So this will help with studying for this. And that will be the end of chapter 26.